Nanoparticles, their design and potential application in medical imaging. Medical imaging focuses on retrieving spatial, temporal, and functional data from physical structures and bodily processes which cannot be acquired without invasive intervention. These imaging modalities are crucial as they make an enormous contribution to diagnosing and monitoring therapeutic treatment of the patient. Nanoparticles are extravagant imaging agents that offer unique biological, pharmaceutical, and therapeutic applications. Nanoparticles can be described as molecules with an average size of 1 to 10 nanometers. That's super small. The size of these particles allows them to easily access the intricate internal body and overcome biological barriers present. They exist in the same size domain as proteins, making nanomaterials suitable for biotagging. Nanoparticles offer many unique properties that may be exploited to refine biological properties and enhance existing imaging systems and create new diagnostics which have only been made possible through the utility of these imaging properties, hence leading to a higher level of efficiency overall due to their adaptability for various purposes. Alterations to size, surface properties, drug interactions such as loading, targeting, and releasing functionalities are all possible to suit the need. They can also be designed to vary biodistribution and aim for a specific target within the body. These nanoparticles are of high versatility. They are also designed in order to attain the best promising detection sensitivity. These nanoparticles must have a very low toxicity level and controllable pharmacokinetic behavior. The utility of nanoparticles as imaging agents such as biological markers, labels, probes, contrast agents, and dyes gives rise to a precise diagnosis and the current state. They also allow optimum visuals of the internal body that are of very high quality, enabling professionals to pinpoint any inconsistencies in both structural and functional dimensions within the body. An active area of research is the development of theragnostic nanoparticles. These are nanoparticles with dual diagnostic and therapeutic functionality. An example of theranostic nanoparticles is targeted cancer therapy. CT, MRI, ultrasonography, optical imaging devices, gamma scintigraphy, and PET are the modalities used in molecular imaging depending on the kind of signal needed to be produced. An active area of research is the development of theranostic particles. These are nanoparticles with dual diagnostic and therapeutic functionality. An example of theranostic particles is targeted cancer therapy. Nanoplatforms. Nanoparticle-based theranostics. The uniquely small size of nanoplatforms enables an achievement of an enhanced permeability and retention effect in targeting foreign objects in the body, such as tumors. What does molecular imaging offer different to traditional imaging methods? Rather than merely reflecting the anatomical changes from pathological tissue to normal tissue within the human body, which would only detect cancers once they have made visible change to a tissue, molecular imaging enables visual representation, characterization, and quantification of a variety of biological processes. However, this is not to say molecular imaging does not have its downfalls, with poor spatial resolution, a lack of sensitivity, and more decisively weak penetration through tissue, limiting the growth and emergence of the technology. Being in the early stage of development, however, molecular imaging modalities are yet to be given sufficient time for detailed research and reconsiderations in its use and design. Nanoparticles are emerging as a new successor to molecular imaging, presenting the major advantages of standard molecular imaging without any major fallbacks. Passive accumulation and active targeting designs are techniques which encompass nanoparticles for medical imaging purposes. Nanoparticles are being developed as molecular imaging agents, reporting the presence of cancerous material or even to the extent of functional characteristics of tumor cells. How do nanoparticles accumulate in tumors? The nanoparticle platforms can passively accumulate in tumors due to the tumor tissue's leaky vasculature and the lacking lymphatic drainage system. Nanoparticles can also recognize and bind to tumor cells through receptor-mediated endocytosis. Nanoparticles' dual functionality in diagnostics and therapeutics allow for their use in targeted cancer therapy, as they are able to detect and examine tumors, as opposed to only being able to distinguish abnormalities from regular tissue. Nanotechnology opens the door to earlier and more accurate initial detection and diagnosis, as well as continual monitoring for cancer patients. Nanoparticles are advantageous to regular molecular imaging in their high signal intensity, large capacity to offload, and their overall stability. Nano-enabled sensors are able to find and circulate around tumor DNA as well as tumor-shed exomes. By way of conventional scanning devices, nanotechnology-based contrast agents offer specific targeting of cancer cells. MRI, PET, and CT scans are most commonly used. Nanoparticles are able to be used as a tracker in tumor growth, but also as an identifier to the cancerous cells in removal of the tumor. This was exemplified by researchers who developed multimodal nanoparticles, which were able to precisely determine and remove brain tumors preoperatively and intraoperatively. Further research in developing silica hybrid nanoparticles had succeeded in clinical trials in delivering both PET and optical imaging contrast in the same platform, with the particles being actively targeted to a specific cancer peptide targeting the specific tumor type. The extensive complexity of design, structure, and application of multifunctional nanoparticles heightens the analytical and manufacturing intricacy. As a result, the recent spotlight and demand for development of the application of nanotechnology and particles in the medical imaging field has sparked billion-dollar expenses on research, manufacturing, analytics, and experiments. These costs exceed $160 billion, significant scrutiny and resource application required to maximize the advantageous practice of nanoparticle medical imagery. Cost-effective alternatives to clinically approved gold nanoparticles, bismuth sulfide and tantalum oxide, are currently of high interest, with aims to achieve the high standard imagery of gold nanoparticle contrast agents at reduced manufacturing and application costs. Literature and studies indicate the necessity for future study and research in the safety of specific nanoparticles. The main question of safety for nanoparticle medical imagery remains: what is the acceptable retention percentage of nanoparticles in medical imaging agents post-diagnosis? The safety of nanoparticle utilization is an intricate balance and combination of the substances, biodistribution, and toxicity. The toxic threat and nominal study of toxicity restrict wide application and accessibility of nanoparticles as a crucial component of medical imaging. Therefore, it is anticipated that the nanoparticles detected by highly sensitive modalities such as SPECT, PET, and optical imaging will acquire a higher success rate due to lower dosage. MRI iron oxide particles have exhibited great success over the past decades, merited by its iron oxide core degradation, with lyosome minimizing retention and half-life. Nevertheless, studies continue to maximize its advantageous application by interest in manufacturing of smaller iron oxide particles, less than the present particles of approximately 60 nanometers and greater. High atomic number nanoparticles allow CT scanning to be achieved at lower radiation rates with better sensitivity and reasonable specificity. However, these particles show minimal advantage in comparison to iodinated X-ray CT contrast agents, as dosage exceeds 20 to 30 grams in desire to achieve higher sensitivity, posing grave health risk and toxic effects due to high retention. Toxicity and biodistribution of the particles encompass the highest challenge to manage nanoparticles as a medical imaging agent due to their size, charge, distribution, dosage, metabolism, and surface, causing physiological disruption. It can thus be concluded that the safety is dependent on toxicity and biodistribution, which are yet to be intricately understood and examined as necessary technology emerges to strengthen studies and long-term experiments. Further improvement and developments are crucial to potential imaging technique. However, control and management of the particles once ingested or injected are necessary due to discrete acceptance of adverse effects of medical imaging agents. Costs of research will continue to escalate as increased interest and demand rises, with each imaging modality requiring individual testing before improvement. While the cost of research and emerging demand is considerable, the potential of application is extensive and vastly advantageous to quality and accuracy of clinical diagnostics, treatment recommendations, and therapeutics.